Can you see it in full screen also?
Yes.
Okay. So where's it? One thing that turns out with virtual sessions that I depend completely on you interrupting me in case it is needed. So feel free to say like, okay, the connection just isn't best, so please repeat this information, and so I'll be waiting for that audio cue.
Okay, yeah, noted.
Yes. So you'll tell me when we shall start. So shall we start?
Yes, of course.
Yes.
Yeah.
Okay. Well, thank you everyone for the invitation for this class. So it was kind of surprised to be invited to these the Bioinformatics sessions for bioinformatics, and I was really thrilled to present it. So for those who don't know me, my name is Angela Garcia. So I'm from Colombia, and I work in bioinformatics for the Global Health Research Unit in Colombia. Currently, I'm a biologist and also a nutritionist, but I did a master's study in computational biology, and I've been having some experience with different classes. So here I am to present you why is bioinformatics important. So why does bioinformatics matter? So thanks for the invitation, and I'll start with maybe something that you've already seen before, but it's thinking around some basic concepts around the bioinformatics or computational biology. Because sometimes in the literature or talking to different professions, you hear both concepts like computational biology or bioinformatics. And funny though, I did found these definitions from a handbook in the NCBI, which describe computational biology more closely related to the development and application of data analysis, while the bioinformatics is more related to the interdisciplinary field of applying computational biology. So it's common to find them both working together and also to being both described at the same time as if they were synonyms, but they're a bit different in what their definition is. So a consideration here is to describe computational biology more in that sense of the development of and application of those data analytics, that development of, for example, the scripts, a algorithm development, like to sit down and think around, okay, how can I analyze this bunch of data and start from this information and result in this processed data? And that always taking into account that it's to understand biological information. That's common between both concepts, that they want to describe biological information. On the other side, the bioinformatics is more in the field of application. So bioinformatics has been around for nearly 30 years by now. And there's a lot of information and a lot of tools that are already there. And a few of them are actually just like, okay, this is the standard one for this process. So in that sense, when we are just using those tools, we're doing bioinformatics. But biology is beautiful. So the thing with biology is that you can find any kind of different problems. And what you will do, it's like for the biological problem that you are actually researching, you can do both. You can use bioinformatics for everything that's already there, but every biological question is different. So when you enter in that realm where, okay, there's nothing here available, you go into computational biology to develop those new tools, scripts, or frameworks that you need. The important thing here is to always have the right tool for the job. Computers just help you, but computers ain't intelligent, even with the AI boom. But right now, bioinformaticians are the ones that decide what do we use, how do we use it. We are the ones that understand biology and pick up the right tool for the job. So in that sense, bioinformatics is really, really, really big. So it takes into account different topics. It's definitely a completely interdisciplinary discipline because we integrate information from different topics, including, for example, computer science, engineering, statistics, but also the more biological, biochemistry part. And it comes together to solve those questions that originally, with just biology or just engineering alone, we couldn't solve. In that sense, computational biology has developed very, very more specific topics, and also each one of them can have a whole class by itself. But the ones that are more listened and we can read around in the literature with different species of animals, bacteria, viruses, are, for example, genomics and DNA and RNA analysis, the structural bioinformatics, systems biology and networks, evolutionary and phylogenetics, proteomics and metabolomics, and of course, machine learning and AI. These are just some small examples, but as I mentioned, each one can be as big as to have their own class of their own to describe what are all the possible available tools in bioinformatics for only genomics, or all the tools for structural bioinformatics, or all the tools for machine learning and AI. And they can be independent from each other, or depending on the biological question, we can all use them for the same project. In that sense, for example, for genomics and DNA and RNA analysis, the most basic questions is like to describe, okay, what about the Human Genome Project? So what are all the genes that are available in the humans, and describe us as we are? And what defines if we are, for example, tall, short, or that we're more prone to some diseases? That's what genomics it's doing. Another example, structural bioinformatics. This one, it's more interested in, okay, we've got a lot of proteins. How do they look in their 3D structure? And how does it work in the organism? So, for example, in here, we've got one protein that might be part of the membrane. So what interactions does that protein have with other cells, with other metabolites? And it can help us to describe the biology. And our examples of systems biology and networks, and this one it's thinking more in more ecosystem kind of way, because like, okay, how, for example, do fungi communicate with our fungi in the roots of trees, in the soil, in the forest? So that's kind of what systems biology and networks analyze, though it can be pretty much worldwide in this case. Another example, evolutionary and phylogenetics. So the most basic and easy example I can give you, it's, for example, the tree of life. So how can we actually recreate the tree of life? And how do we describe that these species are more closely related between them than others? And for example, why do people say that we come from other primates, or we are part of the same group of primates? What are those genomic characteristics that actually groups us together? Other example, proteomics and metabolomics. This one is more like taking a pinch of the information from the structural bioinformatics. So we're also in the realm of proteins and also in the realm of metabolites. So think here by pure biochemistry on the different metabolic routes that how does our body synthesize energy or glucose or all those metabolites that we need to survive. And finally, the other two, biomedical applications, this one is more closely together to, okay, let's use all the previous information that we had. That's more like, okay, let's learn and know how everything works. Now let's think how to use it. So where those proteins or those metabolites interactions actually helps us, for example, for drug development or antibiotics or other different kind of chemical sources that might be of interest in biomedicine. And finally, machine learning and AI. And this one, it's more closely related to the engineering side, but let's think about the problem that biological data started small and there was really a small amount of information available. But right now, the amount of information that we had, it's huge. So there's no way that we can stay with the old school versions that we can all by pen and paper do everything. We need computers to help us analyze that huge amount of data. And when there's thousands or even millions of data to analyze, machine learning and AI can help us to reduce the information and describe what are the patterns or the most important insights that we can find in our biological data. So far, I'd like to present you this article that describes in a really easy way and a nice way nearly everything that I've mentioned so far. I do recommend reading it because it actually is like thinking around like, okay, any biological question that you think through, it can be translated into data that you can use computers to help you get insights into what's the most important information there. And so that's just one of the many reasons for why bioinformatics is important. Informatics offers you multiple tools to solve different biological questions. It is interdisciplinary and it complements nearly any kind of scientific biological study. What you have to actually think about, it's like just starting now and say, okay, I have this biological question. What data do I need? How can I get it? And how can I analyze that? And in that, how can I analyze that? Bioinformatics is key. So in this case, like you want your car, your project in science, your driver will be that biological question. So that will describe everything. What do you need? How to analyze that? So far, any questions?
So please, if you have any questions, so far, kindly unmute and ask.
Okay.
Anyone,
So
Mike,
interrupt
if you have
me.
a question.
No, no questions for now.
Okay,
Okay.
good.
So no questions are assumed. Okay.
Okay, so let's keep going. So far I've been talking about like, wow, bioinformatics, it's amazing. And maybe you can tell I love bioinformatics. But what about some real life examples? And the biggest one that I thought about, it's something that anyone on Earth experienced, and it was the COVID-19 pandemic. So I like to talk about it because we all suffered for the different things that COVID-19 affected our everyday lives, not just for how we move around, how we relate to people, how the news talked about it, But we all had some experiences with it. And through that, what I actually was trying to do was, okay, let's learn more about the COVID itself and how everything went, like all the possible public health decisions, all the information that went to the news, the idea of, okay, getting that exam of if you're positive or negative for COVID-19, all of that behind it depended entirely on bioinformatics. So how did it work? In here, I have some articles that described it. But for example, in the world of the genomics and DNA and RNA analysis, we've got to analyze that. And actually, a nice tip is that the first whole genome of the SARS-CoV-2, it was published by December of 2019 from China, and they just let it out into the world. They were already in their homes. They couldn't do anything else by December. The rest of the world, month by month, we were all inside that same situation. But by having just that first genome, we've got, okay, which are the genes that are here involved? How can we start to analyze what is this virus? What can we do with it? How can we actually fight it? So for example, one of the other visions is to do structural bioinformatics. And in this case, this is a 3D protein for the ACE2 protein. Interestingly, that's a human protein, not a SARS-CoV-2 protein. The SARS-CoV-2 protein, it's the small one, like yellowish one, it's up here, that belongs to the S gene. That comes from the genomic side of the information. And in this case, it was to solve that question, for example, of, okay, how is this virus being transmitted? How does it enter the human body? And one of the main connections that they found was specifically this protein, the ACE2 protein. And ACE2, it's found in nearly any epithelial cells. So that's how they found, like, okay, this is an airborne virus. So we need to use face masks to avoid contact. So this kind of information did replicate in how we actually moved around the pandemics. Afterwards, this systems biology and networks approach, this one was used, for example, to analyze which were the genes that were activated inside the human body, or on another study, what were the different proteins that were available in the SARS-CoV-2, and if there were any drugs that could actually affect them. So in this case, it was like to search for also those drugs that might be like first line, that, okay, for all the available drugs that we already have, what can we actually use to fight the virus and help those people that were already sick? So this is another approach that was there. And next was the evolutionary phylogenetics. So in this case, it was thinking through the idea of doing more like an epidemiological surveillance. And in this case, it was, okay, let's answer the question of, we know that the virus started in China, but how it is being transmitted. So the one from China traveled to Europe and then to Africa, or maybe directly from China to Africa, or from Europe and then to America. How did it go around? So by analyzing this kind of phylogenetic trees, and by grouping, okay, what we have here, it is closely related or really, really similar to the one from Europe, we will say, okay, this one came from Europe. And do that surveillance of, okay, we have to be really careful, for example, at the airports, because that's the exact place where all these kind of viruses are entering into our country. And afterwards, they will just be amazingly good at being pandemic worldwide, anywhere. So this information was to also do a follow-up of the different variants and to see how the virus itself was mutating, how it was changing, what was changing. And this was also the basis of, okay, what's changing, what is not changing, and from what is not changing, maybe create the vaccines that we use for avoiding this pandemic for more years than it was. And the latest article that I wanted to show, it's precisely one that combined proteomics and metabolomics, biomedical applications, and machine learning. And this was to actually try to predict from just the metabolites from the serum in the humans what was the prediction of how severe would be the cases of the COVID-19 in the human. So what they did was to take out different serum profiles from people that they knew that were severe and non-severe, analyze the proteomics and metabolomics, use machine learning and AI to actually group that really huge amount of information of the whole serum information of the metabolites that were present, the proteins that were present, try to group them into which one were descriptive or not of the severity of the disease. And finally, with this, get some key metabolite that you could actually go and check in the serum to check, okay, this human might be one that goes into severe cases, so let's prevent anything further and take decisions. So in here I have this in slides a little bit bigger, the information that I just showed you. So this is one from the genomics, the structural bioinformatics, and the ACE2 protein. This one's a systems biology approach, for example, to treat combinations of drugs and to use in the clinics. Phylogenetics to track transmission, and the proteomics and metabolomics approach. So I like this idea so far because COVID, we experienced it all, but it's also to think around the idea, okay, what are scientists doing? How can we actually fight that? So how do we fight different infections? We try to manage them with drugs, we tried with the vaccines, we tried with all the use of the face masks, and they all had different scientific information. And as you can see, they were all obtained thanks to bioinformatics. In detail, let's go a little bit further, a little bit step by step on what happened. Let's go into the phylogenetic analysis. So this is another article that did describe that, in essence, bioinformatics is behind all of the COVID-19 pandemic. And for example, in here, what we actually could go and search for information. I can start from just extracting the whole DNA or the viral DNA in different individuals that were infected. Also, do a really accurate metadata collection. So, what kind of individual was, when, and where all of this information was captured. And from here, the viral DNA, you can actually use that for all this analysis, compare different sequences like human 1, human 2, human 3, human 4, and also against the 2019 genome that was available from China. Compare these sequences and search for mutants. And those mutants will describe this, okay, this one's similar, this one's different. And actually, from these mutants, there was also an additional step that was to classify the mutations. And I don't know in your news, but in Colombian news, it was common to have nearly 5 minutes on each news session that will describe, okay, today we've had so many infected people, so many deaths. And the main variant that's around, it's the, for example, Omicron or Delta. Those names comes from this mutant classification, but as happens with the news, not all the available mutants go into the news, only those that are of epidemiological interest. So all of this just went into help nearly every country to actually do real-time epidemiological surveillance and make epidemiology-based decisions. This one's taken from the UK, which they had even more sequencing for the follow-up of COVID-19. And this is an example of how it looked worldwide. So what kind of information do we have here? Like the geographical and timeline data, and also whether all the different variants that are available. As you can see in this mesh of colors, there were really a lot of variants, a few more successful than others. And in the phylogenetic tree, we can see here how are all of them closely together. Though there's a little bit of a bias here because the tree will mostly show that UK had way too many sequenced isolates compared to the rest of the world. And so far, up till now, I'll stop here with COVID. So, any questions?
Yes, ma.
Go ahead.
Thank you very much, ma. For the variants, I want to know, is it possible that the body of the host can affect the can like cause a mutation in the organism? For example, say COVID-19, it affected two different people. Could it be that the individual themselves could affect the type of variants in which the isolate will end up being? Thank you, ma'am.
That's an interesting question. I didn't look into information around that, so I'm not 100% sure. But what other articles showed that, for example, well, the viruses can mutate on each individual also. So, for example, that's a mutation that you mentioned that it's in two different individuals. In one, if it had, for example, a really, really good immune system, the variant might just die. While in the other individual, if it has a weak immune system, it might actually develop into accumulating even more mutations, and that those additional mutations then transmit into other people and might have that possibility of affecting other people. So it do behave really, it could behave differently in different individuals. Because in here it's also that question of, okay, how does the immune system work against the COVID-19? I don't have experience about it, but I know that it could be really different between several individuals or even populations.
Thank you, ma'am.
Okay. Nice. Any more questions? Think.
I guess not. So I think we can continue.
Okay. So let's keep going then. So the next part of my talk would be, okay, we started like with some brief definitions, why is bioinformatics important, and a real life example. So let's think in more detail. So how can I actually use bioinformatics? And in this case, it'll be focused on a story of antimicrobial resistance studies, which is the ones that I've worked with in a more close way. So what's the problem? So you mentioned before, we start with the biological problem. So what's the problem of antimicrobial resistance? Right now, the antimicrobial resistance, as you can maybe tell by the name, is that some microbes are resistant to antibiotics. And this is not something that's from nighttime to daytime. It happens from an accumulation of factors because bacteria do have very, very, very different populations worldwide. We have some populations in our guts. We have other populations in our skin. There are different populations of bacteria in the environment. But when there's some infections, well, we use antibiotics. But when we use them, we're actually eliminating everything that it's susceptible to those antibiotics. But as chances can be, there are some bacteria that have the mutations or the genes to actually be resistant. If we do not use correctly the antibiotics, what will happen is that those bacteria will still be alive there and they can thrive. Nothing else is there and no other competition. They can just have all the nutrients for by themselves and just grow. And because they're resistant to antibiotics, well, some issues might arise when we keep on having all of the different antibiotics. So this can come from different settings, from the prescriptions that we have with the different doctors, or maybe directly at poor infection control in the hospitals, or maybe the lack of hygiene and for sanitation. So water and soap is really, really helpful. Or even if some animals got those bacteria and the antibiotics weren't used correctly in the animals to actually eliminate those bacteria, we can afterwards eat those bacteria if we're eating that those animals or the different fruits or vegetables that can have in their surface the bacteria. So it can come from anywhere, from the water we drink, from the foods we eat, from our neighbors, or from our visits to the hospitals. So in this case, it is spreading fast throughout all of this possible change, which is currently described as the One Health perspective. So to think around the idea that we're humans, but we're in an environment, and in that environment, we're actually interacting with other people, with other institutions, we're eating food. So from any of those different sources, bacteria can come and colonize us and even infect us. So what's the idea here? That the problem is that we need to understand that antimicrobial resistance. So how does it develop? How does the bacteria actually, how can we actually say, okay, this bacteria is resistant or this bacteria is susceptible? So the idea to understand that is to actually go through a really, really tough scientific analysis and to, okay, we can use epidemiology here for that surveillance and study those patterns, causes, and the effects of antimicrobial resistance throughout the whole chain. And in this case, what we want to see is, okay, if we're talking about antimicrobial resistant bacteria, well, we will first look at those bacteria. We won't look for fungi or we won't look for viruses. We will look specifically for those bacteria. And what we want to see is, okay, how's the biology of that bacteria? How can it grow? How can it spread? How can it infect us? And in that sense, what we might be well fragmenting here, the problems, okay, who is it affecting? In this case, humans, animals, or the environment. We need to be really wary of where am I going to find information? Am I going to study only the humans? Am I going to study only the animals? Am I going to study the bacteria from the environment? I can focus on only one of those at a time, or I can try to analyze them all. So who's, in this case, the one that I'm going to study? In most cases, it's humans, and I can focus the rest of the talk from a human analysis. What are we studying? The bacteria. Those are the, I don't know if it'll be right to call them enemies, but well, they're the ones that we don't want to be zooming around and being there and infecting people. We want to study those bacteria and those bacteria that are there ready to infect us. Next comes the how. So how is actually a bacteria being resistant? And in this case, the best way to actually get that answer of how it is happening is with genomics. All the data for that, it's in the DNA of this bacteria. And lastly, when? Because it's important to know in space and time what's happening. So I'll go back one step here. And for example, for COVID-19, the pandemic's over, luckily. But in this case, COVID's still there. It behaves more like a flu now, but in that space and time, we can think about that, okay, the timeline, which was of most interest for everyone, was that specific period of the pandemics. Right now, it's still there, but in time, we might see some variants that are still infecting us, but are not as problematic as it was back then. And in space, well, it's different the behavior of how it goes inside the clinic, how it is in the house, how of your family house, for example, how it is in a completely field of, for example, fruits and vegetables. So the space and time is also important here. So what do we do? To understand and do a real, as close as possible follow-up of the antimicrobial resistance in long version and step-by-step here, what we start with is, okay, we have to get the data. From where? From collecting the samples. And as you see here, I'm using the same icons because we start by collecting the samples within the humans, the animals, or the food that we're eating. From those samples, we not just take the samples and that's it, but we also take all that metadata of interest about the space and time so that we know, okay, this sample was in this moment affecting these different individuals. So really important to have really, really good sample collection alongside the metadata. Now that sample goes into the laboratory and the lab people, microbiologists mostly, start the evaluation and think, okay, from this sample, let's just take all the available bacteria and describe it. What species do we have and what is their resistance? That is the antimicrobial susceptibility testing. So to actually describe, well, these ones are resistant, and specifically to which antibiotics are they resistant. Afterwards, then, after confirming all the previous information, we can say, okay, we want to go further and we want to know from the genomics what's there, what does actually give it the bacteria the top resistance characteristics. So DNA extraction, it comes from those bacteria, and the DNA, it's a huge library, which in theory holds every single one of the information what that bacteria can do. So from that DNA extraction, we're still with some vials, liquid vials. So if computers are going to help us, we need to translate that for the computers, and that's the sequencing. So sequencing takes that DNA and transforms it into the letters, and from those letters, we will start with the bioinformatic analysis. And after all the possible bioinformatic analysis, we get into finally describe, okay, how is this bacteria resistant and why? And report back what's going on here. So hope you're not lost here. I'm going to make it a little bit shorter and brief. So in this case, the first part, sample collection and metadata, that's mostly going on in the different institutions and all sites, and with the different professionals, mostly in the public health area. They send that information to the lab, so all yellow goes on in the microbiology lab, and afterwards, there comes the bioinformatic analysis. So everything is already digital. Let's play with it. And this is, in essence, what is going on in the whole GHRU project. So information from the different institutions and health professionals, all the work that the microbiologist in the laboratory does, afterwards all the bioinformatics analysis, and finally, by taking together all of that information, we get the results, and to actually describe what's going on with that biology of the antimicrobial resistance in that population specifically, and take some possible decisions for public health institutions to apply, for example, new rules or new information that will help them treat and prevent antimicrobial resistance afterwards. So, of course, this is a bioinformatics class. I am a bioinformatician, so actually, each one of these steps can take up some time to describe them in detail, but I'm going on to describe in more details the bioinformatics part specifically. So, for bioinformatics, we start with basically 4 letters. The 4 letters that define the whole DNA, A, C, T, and G. Those are our 4 letters. So for engineers, it will be binary code. For us in genomics, this is our main language, those 4 letters. Now from those 4 letters, and by finding out what the order of those letters in our bacteria are, we will try to find some biological insights, and specifically in this case, to describe the antimicrobial resistance. So keywords, these are the 4 main steps that are being made during bioinformatics here. But the first one's more from the lab because it's sequencing, but we need to start to actually get those letters. Afterwards, assembly. Then with the assembly, which is like having the data in a more combined way, you get to do the gene annotation. Then afterwards, phylogenetics. And don't worry if this all sounds a little bit like, ooh, too many concepts. We'll go one by one, slowly, to describe them. But the idea is think around, we start from just DNA, and we will try to get some biological insights on antimicrobial resistance. So the DNA, just to recall from what you already seen before, but it's a DNA strand that's formed by the A, T, C, and G with the pairs from CG and AT that they actually work together. 18 the double screen and double and forget the name in English. Oh, the H bonds, double H bonds. Well, the CG is triple H bonds. In this case, this actually helps us because in bioinformatics, one of the things that's important is that the double helix, because of its complementary, we only work with one of it. So you will always see like this is the DNA, but only one of the sites is actually the letters that we're working with. Why? Because the complementary, we just can find it from the complementary information. It's an additional step maybe to calculate that. Yes, but it's okay because one of the issues with the DNA is that DNA can be really, really big or small. It comes from just every single cell of the bacteria, but just one bacteria can have around 5 million of these letters. There are some bacteria that are smaller that can have like 3 million or 500,000, but there are others, like for example, Klebsiella, that can have 6 million or 7 million. So imagine this. We want to get all of the 7 million letters for that DNA of a bacteria. That's a lot of information. So how do we actually get it? So starting with the bio, with the DNA that was from the bacteria, how we actually get the letters in the digital form. That's sequencing. And there are several different possible sequencing technologies, but the most common one is Illumina, and the one that I'm bringing here. And Illumina sequencing, what it does, it takes up all of the DNA from the bacteria, it shatters it and fragments it into small fragments of around 100 or 150 nucleotides. And those small pieces are then added an additional DNA on each side, and that additional DNA works for two things. One, to know that DNA from which sample it came from, and second, to actually aid with the sequencing afterwards, because this it's DNA that will help attach it to a platform where all the magic of sequencing happens. So this extra DNA, it's called an adapter. And in Illumina cases, what's going on with that small DNA is that, well, one small DNA will be really, really difficult and hard to find. So PCR here, amplification process happens, and from one piece of DNA, we get multiple copies. And with that multiple copies, we can actually start to get a visual of which are the letters that are going to be there. So afterwards, that we already have like our library prepared, sequencing happens, and for each different letter, we've got a color. So A's in this case are red, G's are yellow, T's are green, C's are blue, and a really, really powerful camera, what's going on is like, okay, in our platform, we've got different sets of the DNA, and each DNA, we can see it from above, and for each letter, it will pick colors. And it will look like this colorful picture, or maybe pixel art. But each one represents a letter, and each letter will be like, by time, a different color, and tell us, okay, this sequence, it's like A, T, C, G in this order. So funny story here is that a few years back, like around 10 years back, the data that Illumina actually sent us was this, all of these colorful pictures. That was really heavy. And afterwards, it was starting analyzing the informatics data. So transform these colors into sequences. Right now, Illumina learn from their ways, and they do it. So they transform these pictures into the actual article reads, and those reads represent the letters of those small fragments of DNA that started the whole. So lots of steps, but in general, we start with a shattered genome of small pieces of 150 nucleotides and end up with those 150 nucleotides, but in their letters. So we now have a digital form of A, C, T, or Gs. And it looks like this. So the output of the sequencing is actually the first format in computational biology that anyone sees. That's called the FASTQ format. So this format, it's in essence what you have in your screen right now. It's a 4-letter format. The first 4 lines format. So the first line, it's for the identification, and this long sets of really awful numbers that you will say like, ugh, I don't want to remember that. And those numbers in essence what just represents like these colorful dots were on a coordinate in the plate. So these are the coordinates. Debug them to remember them. The one only number that you might even look at, it's this last one, but it's a 1 or a 2. That's it. Because in FASTQ file format, the FASTQ for Illumina, they had a thing that the sizes that they manage are that 150 nucleotides, but they did a trick and try using longer sizes, like around 300 nucleotides. But what they did was, okay, let's sequence just this first part on one cycle and then the second part on another. And because this is all shattered random, we can cover all of the information of the genome at a time. So all of that confusing. Why do 2 cycles? It's what in bioinformatics, it's then described as the pair-end sequencing. And what you see is that for one sample, we don't have one file. We've got 2. One for the forward, another one for the reverse senescence. When do we see that? This number, if it's a 1 or a 2. That's all you've got to remember from that first step. Then the second line, it's the nucleotides on itself. As you see, here are the A, C, T's, and G's perfectly in order for this small fragment of around 100 nucleotides. Next goes the third line, which is always this plus symbol. And that's it. Sometimes in some files, they will repeat the ID, but it doesn't have anything else to it. It's just there for the format. It's like, okay, as when you're saying like, okay, a reference format and it's first the name, then a comma, and then the last name. That comma's there for the format. So in here, that plus, it's there for that same reason. It just exists for the format. And the last line, it's the quality. And the quality in here, as you see, that's a lot of letters and symbols. But in essence, what they represent, it's what it's called a Phred score. And that Phred score underneath represents what is the accuracy of each one of these nucleotides being correct. So I don't remember each letter, but for example, the G letter represents like a 1 in 1,000 probability of error. So yes, we can be sure that nucleotide is correct. We don't actually know them all. Why? Because this is something that computers can help us, like, okay, this is in that just one letter Phred score way to say this is a good or a bad quality. You remember it? I will just analyze it. So there it is. What's the thing here? That you will have as many quality letters or symbols as many nucleotides you have in here. So these two should have the exact same size. And as you can see, this is just one sequence. From what we had from the Illumina were millions, or at least thousands, but usually millions of sequences. So this file, it's just those blocks of 4 letters repeated one after another, each one representing a different sequence. So I hope you're not sleeping up, but right now then, this is the first format. So what's the problem that arises here? We fragmented the DNA. I mean, we've got a really beautiful whole genome when we had the bio, but for the technique purposes, we've got to shatter it. But we want the whole genome. We want it complete. So this is like thinking around the idea that, okay, we really made a really, really big and, let's say, complicated puzzle. So how can we actually take each one of these different pieces and recover the puzzle? So that's the next step here. So we've got to take those reads and try to take into account that because this was all shattered random and we didn't have the information for just one bacteria, we got the information for multiple clones of that same bacteria. So we can have by random chances that some bits of information might be overlapped. And with it, we can get context and afterwards with other efforts try and recover the whole genome. So from the actual Illumina, for example, MySeq platform, how do we actually get that puzzle? That's when we start to build the puzzle with the assembly pipeline, or actually trying to get that assembly. So first sequencing, now assembly. So assembly in essence, or in an easy way of understanding, it's starting from that FASTQ format, use many tools which will guarantee the quality of what we're actually looking for, and end up with an assembly in a FASTA format. So think around this like, okay, what about if you had a beautiful cookbook in your home and just shred it in multiple pieces? Those are the FASTQ. What we want to do to recover that book, to have all of the information there as it was in the beginning. Though there's some really troubling issues that might arise in that transformation because, for example, well, there might be some bits of words that are unique, or maybe some colors, or maybe some markings that you will say, oh, this is exactly this page, so I know exactly where to put it. But what about other pieces, like, for example, the margins, that they can be all blank in any book? So I don't know if this is from page 1, from page 10, from page 100, no idea. Those are kind of those issues that arise during this process. So we attempt to recover the whole book, but at most we can recover some recipes, but that's enough. So what is an assembly? The FASTA format that I mentioned before looks like this. So in essence, this is like a shortened version of the FASTQ because it only has 2 lines. In this case, the first line is the header, and the second line is the sequence. So header, it will be the description of the sample. Say this is sample 1, gene 1, or fragment 1, because we don't really know if there are genes or not there so far. We know that might be from this shredded piece. Okay, this might be book 1, recipe 0, for example. And this is the whole sequence that, or the context that we could recover. So this assembly, or this FASTA format, ideally, in taking into account the biology of the bacteria, if we've got one bacteria, it will have one main chromosome, maybe some plasmids, but if we're just assuming this is just one big chromosome, then our FASTA format should have only one sequence. What happens in real life is that from one bacteria, we're happy if we have around 90, 100 contexts. So not one, but 100. Still, it's enough. As I mentioned before, it's like thinking around the idea of, okay, I can recover the recipes. It might not be the whole book as one unity, but with the recipes by itself, I can work it. So the idea with this is that the assemblies are evaluated here for how contiguous, how complete, and how correct they are. The contiguity here is to think around the idea that the best contiguous sequence would be the whole chromosome, but it is okay to have just bits of information for each one of the pages, for example. The completeness, this is an evaluation that's actually a cross information with biological data, because we've got to think here, okay, did I really recover the whole information from one bacteria? How do I actually know if it's there? So one of the ways of looking at it is thinking like, okay, if I am, for example, a whole human being, I will need to have eyes, legs, and well, maybe that's a really bad example actually. Discretion. Um, so in the case of bacteria, you will need to have all of those genes that will be essential that any bacteria can live without them. So all the core genes, so things like they've gotta eat, they've gotta create energy, they've gotta create the membrane for them. So those genes will have to be there to actually describe that I do have the whole information from just that one bacteria. So that's what the completeness looks for. And lastly, the correctness. This one is to think of the idea that we want as best as possible that all the information is perfect, but sequencing platforms ain't perfect. I mean, that's why we've got the quality information. There are some errors there that could be. Gotta be sure that these changes in the nucleotides are actually real information or avoiding any possible technical errors that might arise before in different sequencing. And so far, that's the idea. So we get the assembly, maybe a fragmented assembly, but as best as we can, as complete as possible, and as correct as possible. And with that information, what are we going to do? So with that assembly, in essence, what we think it's there, it's that it started from the DNA. So we do have a representation of all the capabilities of that bacteria inside of that assembly. And assuming here and using the central dogma of molecular biology, if I do have all of the DNA information, I have, in essence, the cookbook, all of the possible recipes of which are the capabilities of that bacteria to do things. In this case, for example, be resistant or non-resistant to antibiotics. So what's the next step here? We've got all the data there. It's a book in which it's already in the same 4 letters, but we can understand what's actually there, which are the recipes. So next step is gene annotation. And the idea with this is, okay, let's understand the genes first. So the basic units for any living thing. And in our analogy with the cookbook, these are the ingredients. So the ingredients for any recipe, and there are different kinds of ingredients. So for example, those ingredients that you just peek at a recipe and just say like, okay, this is really, really unique of that recipe. The one thing that pops up to my mind is, for example, for Americans in December, they love turkey. So what about a recipe with turkey? They're not just for any time of the year. They're just for that Christmas Eve recipe. So for Christmas Eve, turkey. So if I find the word turkey, that's my ingredient of interest. In this case, if I find a gene that's specifically for any resistant, that's the one that I want to find. So that first type of genes, which are the ones that are simple. So if I find the gene, then most likely I'll have the protein. And that protein, it's the one that's in charge of maybe cutting the drug or driving a direct antimicrobial resistance. What's the second type? We've got some point mutations. So mutations in these cases, it's for genes that are there. For example, it might be even core genes. But those genes, with some changes, they will, for example, not allow the entrance of the antibiotic to the cell. So in a wild type way, in a normal susceptible way, the gene is there, but the drug can enter the bacteria. But because of a mutation, maybe that drug now cannot enter. So that's another type of ingredient that I might look for, but I need to be careful with how to handle that. And the most complicated ones are those that are regulating other genes. So in this case, it's like those kind of ingredients that might be present everywhere, but depending on how you use them, all of it changes. For example, the amounts of sugar and salt that you add to a recipe. So if you add too much sugar, you'll have one recipe. If you add more salt, you'll get another recipe completely different. And those are things that are pretty common in any recipe, salt and sugar. I mean, how can I actually see that the amount of any of those or the appearance of both together will regulate that antimicrobial resistance? That's a really big challenge. So most of the time, what we're looking for are the first ones, the simple ones, and at most the medium ones. The third ones, nearly always, it'll be like thinking around, okay, this is a new biological question we might want to ask and leave it for another project. So how do we know that these genes are effectively related to antimicrobial resistance? So we're not doing this from scratch. There's actually a lot of information from different databases that are already there with all the available information about what the name of the genes, the information of the genes, in which species they appear. And for example, in here, CAT2, the CART, the Comprehensive Antibiotic Resistance Database, or there's a smaller one specifically in the NCBI that's called the NDARO, National Database of Antibiotic Resistant Organisms. And both you can all check for those genes that are already looked for in different literature or in different organisms and in different labs that they actually confer antimicrobial resistance and to which antibiotics it does confer that antimicrobial resistance. So what's the key information here? That those databases, it's previous information that can help us to know which are those genes of interest. And the importance of databases of itself is that you can actually search information in them. They're already curated. I mean, there might be errors because humans, we do have errors, but the information is there as complete and as best as possible to say that, okay, yes, this is confirmed that it's a gene for antimicrobial resistance to, say, carbapenems. And the good thing is that they're most of the time on the internet, so you can, in here, have the links to actually explore any of the two databases. So I've talked about databases, but where else can I find information? So there are three really, really, really big databases. That's from a consortium here, the International Nucleotide Sequence Database Collaboration. That it's a huge collaboration between Europe, Japan, and the Americas. So the DBG from Japan, the EBI from the European Bioinformatics Institute, and the NCBI, which is the one in the United States. So if you look for information in each one of them, it can connect to the other two. So you can have information here about genes, about proteins, about samples, about projects, about lots of biological information. So what is database? Why it is so important? We're not just gathering a lot of biological data like, okay, let's just file it there as it is. Data, it's input there in a structured manner so that you can have as best the most information possible for just one record. And think here a record like when you add a contact in your mobile phone, say a family member, a friend, a co-worker, for each one of those records, you don't just add the number and that's it. With just the number, I don't know if you have that issue that if I forgot to add the contact, I'll have that number and I'm like, who's this? And that's the problem. Who's this? When you added more information, it will be like, okay, now this one has a name, a surname, maybe an affiliation of which institution do you know it from. That actually adds up information on how you handle your conduct. So next time you get that, it won't be the number, but the name. So you'll say, oh, yes, it's this people I can talk to about this and this and this. And if they're someone that just lost the cell phone and say, oh, I changed my number, so you can edit that and modify it. That's what the database does. And that's what happens to genes also. So maybe a gene was reported 20 years ago and had a name, and someone on the other side of the world found the same gene but named it differently. But then when they compared it, they will say like, uh-oh, this is the same record. We need to make it just one and keep this one name and keep all of that information as curated as possible. So that's the key information with databases. So to be as best structured and complete, to give you the best biological information available. So what information do we have? So we have information about antimicrobial resistance genes, which those are that directly describe the resistance to antimicrobial drugs. We also have virulence genes, which are those that come from the bacteria with those advantages to be the first pathogen to arrive at an infection site, for example, or say these are the ones that are more competent and say like, this is mine and this is my territory and I'll be here and nobody else will bother me. Those are the virulence genes. And lastly, the typing genes, which these typing genes are more closely together, for example, for the classification of those mutations in the COVID. So for bacteria, we also have some subclassifications that will help us describe those populations and say, okay, this is a population that belongs only to this country or only to this clinic or only to this geographical area. And it's of interest because they have to be kept as best there. We don't want them to spread. So if they're just in that tiny space, that's good. But how do we know that they're all in the tiny space? Because we're following them up with those typing genes, with those subclassification. So this subclassification will be like saying these are different groups in our workstation. So group 1, group 2, group 3. In essence, that's it. So these are all the genes that we're looking for. For each one of this, there are different tools to actually find them. But they all depend on the idea of having a previous database. So you can compare my assembly and say, okay, my assembly with the database. So in my assembly, which of all of these genes are actually available here, and list them. So we will say, okay, now I've got a whole bunch of antimicrobial genes for my assembly, another bunch of virulence genes, and it can be subclassified, thanks to the typing genes, into this group. So what else is there to take into account? So some genes come from ancestors and others from neighbors. Bacteria are quite social. So from ancestors, from clones, they can duplicate their DNA and give it into their their daughter cells. And in that sense, the same information that the parent cell had, the daughter cells have it. And in both cases, when we compare them, we can say like, oh, they're identical. But what happens? There's some cases in which bacteria talks to neighbors, and in that they share information. And that shared information can also be some antimicrobial resistance genes. So how do they get it? So that's, for example, the cases that I mentioned before. Like, we expected one huge chromosome for the bacteria, but what about if there are plasmids? So now we've got ideally one chromosome and one plasmid, so we'll have two sequences at least for that bacteria. So plasmids get a lot of information, and they appear mostly by conjugation. What do we have also in these elements that can be transferred between bacteria? The transposable elements, and these ones are mostly from different phages. So phages can add or remove some information and transfer it to another bacteria. But they'll have to have those sequences that you will say, okay, I can start from here and end here and remove this part and transfer it all to the other bacteria. So we will look for also those transposable elements, which can also be found in databases. And lastly, for example, some incompatibility groups in which we're thinking these are big chunks of DNA that can be transferred from one bacteria to another. And sometimes this occurs, for example, during transformation, because, well, the bacteria dies and all of its DNA is just there. So another bacteria that might be passing by just by luck can use that DNA and use the genes that were there. And with that, gene annotation can be summarized in taking into account that just one bacteria can have multiple sets of information. So we can have the AMR genes, the virulence genes, the typing genes, and also all of the information of the mobile genetic elements. And so this kind of information just gets adding up. So lots and lots and lots of information. Just for one bacteria, we've got all of this. But we don't want to study just one bacteria, because what was the original problem? We're thinking about how AMR is being transmitted. So we want to compare, for example, that the bacteria that someone on a room at a hospital had is the same one as the same patient in the next room or on another unit inside the clinic, or if it's the same one that's outside in an animal, for example. So I've got to compare the bacteria from the human to the bacteria to the animal, or the bacteria from the floor 1 in the clinic with the bacteria in the floor 2 in the clinic. I at least need 2, 3, or even more. So that's the next step. But just to crush that, in real life, this is what one bacteria might look like. And for example, well, this is the bacteria. We've got in here some antimicrobial resistance genes from that simple possible information. The effects pumps here, for example, the mutations, because mutations in here just allow them to say like, ooh, this is an antibiotic, spell it as fast as you can. Virulence factors for biofilm, toxin secretion, for motility, Flagella, Fimbria. It's like thinking also in the idea that, okay, they can pick up like, oh, there's an antibiotic here, run away. If they can run away, well, they won't be able to be eliminated by by the drug. And so one bacteria, many lots of information. So what's also the catch here? Do bacteria behave the same? So the answer is no, because bacteria do change with time. They both can transmit or earn or give up genes. Also, some of them can be from their parents, others from neighbors. Also, taking into account that through time, we can they can adapt to new environments, and across different populations, they can kind of rise by time. So we can start to think like, okay, is this bacteria from the human and the animal the same? If they're not, so how they're not? Are they different populations? So all of this is going to be analyzed with phylogenetics. And phylogenetics then, it's more of a population and evolutionary analysis, but in a nutshell, it's just to think through the idea that we've got the assembly, we've got the sequences. We can look after which ones are similar and group them together, which ones are different, and say split, separate, go with your peers. And what's the idea of going with your peers? Each one of these different I forget the name, branches. The different branches may describe characteristics of, okay, this population came from a bacteria ages ago that did have this gene and it was transferred from mother to daughter on different generations. Or maybe this tree is not as easily together and there's a really weird one here that appeared out of nowhere, and maybe this one it's like, okay, you earned it through another mechanism. So phylogenetics, what uses it's that we group what from genomics look as closely together as possible, but there's a catch here and a careful interpretation. We are assuming that similarity comes from ancestry to create the phylogenetic trees. So sometimes if, for example, a gene, it's just one that it's jumping across different species or groups, this phylogenetic tree starts looking like, why is they together? This group doesn't make any sense. It might be because of that, because the gene is just jumping across different species or different isolates. So here on starts like an additional steps of analysis of results. But so far, that's as best as we can in bioinformatics sometimes, and it's in Resume. It started with the millions of bits, that those puzzle pieces with only 4 letters. We recreated as best as possible the assembly, and then annotate the genes and describe as best as we can using the databases what genes are there, what's the antimicrobial resistance that genes can actually describe, and afterwards use the population analysis with phylogenetics to study the overall population. So are these bacteria similar, how they are, how they're not. And with it, try and describe as best as possible what's currently happening with AMR and transmission, and from this, take up some decisions for public health policies or other possible research investigation that can aid us into controlling the antimicrobial resistance problem. There are some additional tools that I wanted to show you, and this one's from the GHRU team, on the CGPS, the Central for Genomic and Pathogen Surveillance, which, as you can see, looks similar to what I've mentioned before for example, the COVID. It's the idea of, okay, what kind of data do we have? We've got the metadata, we've got the lab data, we've got the bioinformatics information. Let's put it all together and to try to analyze what's happening here. So this is from a real article from my country, from Colombia. And what we have here is this tool called MicroReact, which you need 2 things. You need a CSV file with all of that merged metadata, lab data, bioinformatics information, sample by sample, and you need the phylogenetic tree. So CSV, phylogenetic tree. And in here, you can see that you've got the when, so where, the geography, when the timeline, the phylogeny, and all the possible metadata with all the bioinformatics and lab information of interest, and to try to describe, okay, are these groups of interesting bacteria or not? And well, of course, for this, you will have to already process everything else that I mentioned during this session. But if you don't have any idea of how to do that, there's this other tool. So Pathogen Watch. This one will actually help you with the analysis itself. So you can just start with the FASTQ files or the assemblies, the FASTA files, and FASTQ files, add the metadata, and it will help you to build the tree and to analyze all the AMR genes that might be present. This is called Pathogen Watch, and it's mainly used for, as its name suggests, pathogens. So it's not for every single possible bacteria, but specifically for a list of pathogens of interest in infections that you can analyze here. But it's a tool that the CGPS offers for you to help with all of your analysis. And I think that we're reaching the end of my presentation. So for the future, what more can we learn? So bioinformatics is actually generating and analyzing a lot of data. Think of it as a really, really big collection of tools that you want to use when the moment is what you need, depending on the biological question. That's your driver to describe which are the tools that you need for analysis at a specific moment. So bioinformatics can help you with that, with analyzing all that data. The genes analysis and everything here will have its limitations. So one thing that I might have described as best, but there are also limitations here. Like we depend on what's already there, but we can think about new ways on a more computational biology ways. Okay, this is the limit. How can we go forward into new things? So computational biology for new things. And in here with bioinformatics, just extract as best as we can the most information that we have. We're still learning. Definitely, it's something that it's always a curve, a learning curve. And there are multiple details to be unraveled. I mean, we have this issue with knowledge that, okay, we want to know this, and by the time we finish and actually find that information, it's like the whole world is just expanded and we have more questions than answers. But that's pretty cool about this. There are also other tools that you can use to keep on answering those questions and generating even more questions. Of course, in these cases, you may have seen, so we do have a lot of interdisciplinary approach, and in bioinformatics, that's key. I mean, these are the tools, but we've got to give the tools for the right job, but the right job, actually, the ones who knows it better than us, it's, for example, in the antimicrobial resistance world, the ones that are in the clinics, public health professionals are the ones that are more closely together to that reality of, okay, people are in here getting infected and aren't being saved by the antibiotics. They're the ones in that first line, and they're the ones that need this kind of information. So we need to engage with them to actually learn more about their approaches, what they need, and how can we give them the information that they need to go and save people from antimicrobial resistance. And that's, well, finally here, the idea that, well, in bioinformatics, our challenge is to transform that data into biological insights and actions to finally reduce the spread of AMR. That's our final goal there. And, well, one final bonus here is that from the Global Health Research Unit, there are also more tools that you can overall use for different AMR research. Well, this is designed for AMR research, but as a tool, you can use it as best as you can for other things. And for example, here, for a really, really engaged data architecture and to think of the idea of what I said, but start with the biological question, then describe which is the data that you need, and afterwards how to analyze that. So we start with the question of AMR research here, but how to collect the data, that's a tool. There's an additional tool here that's called EpiCollect that will help you, for example, to collect that metadata information. But this is not focused solely on AMR research. You can adapt it to other metadata collections. The data from EpiCollect can go into Pathogen Watch or into Dataflow. And Dataflow is another tool, but Dataflow what will help you with is, for example, in that step of merging metadata, lab data, and bioinformatics data, how to actually make it smoothly together merged data flow. And the information from Dataflow actually can give you, for example, the CSV. That's the main key, one of the main files that you need for MicroReact. And all of the information in here, the last tool, it's the AMR Watch. And AMR Watch, it's similar to Pathogen Watch, but specifically to describe as best all the information available for AMR research. So, and it might be a little bit of a lot of information. I hope that could explain it as best as possible. So, any questions?
Yeah, thank you very much, Angela. So we have like some time for questions. So Esther, you can go first. Then Victor, next.
Okay. So I will try and look for them at the
Thank you for
chat.
the session, Ma. I have
You're
two
welcome.
questions, and the first has to do with the movement from sequence to assembly.
Mhm.
So the question is it just one string or is it a sample that is sequenced? Because when you mentioned assembly, I was trying to figure out, okay, from is the genes that are being assembled, are they from different isolates or is it from the same isolate? Since it's you said there are billions of sequences. So are they fragments of one string or are they from different strings in a sample?
Okay. I might need to clear up two things here. The first one is that during sequencing, they actually merge together and even mix different samples, but the Illumina actually separates them to tell you like sample 1, sample 2, sample 3, sample 4. For your question, those FASTQ files that got millions of reads, that belong to only one sample, one bacteria. So the process of going from the FASTQ file to the assembly, it's still from that single bacteria. So you've got to do that step for each sample separately.
Okay, thank you, ma'am. Then the other question is the assembly is just to bring other fragments of the same bacteria into one sequence. Is that what the assembly is about?
The assembly ideally will try to do that, yes. And in essence, it's assemble all of the reads from the FASTQ file into as best one sequence.
Thank you, ma'am.
Mm-hmm. No, thank you for the question.
All right. Good evening, good evening, everybody. Thank you very much for this presentation. So my very first question is, when we're talking about movement from DNA to epidemiological surveillance, you talked about metadata being collected. So I want to ask, why do you collect this metadata? Like, what is the actual need for collecting it? That's my first question.
Okay, good question, because it wasn't as clear during the presentation. Because think in the AMR that well we're focusing mostly on the bioinformatics information. This is part of a project, so you've got to always have your biological question as the first thing. And our focus was to find specifically all the possible genes that might explain the antimicrobial resistance. They're not just there. I mean, it's not just like, okay, I've got a bacteria that's resistant. No, that bacteria comes from a human being or an animal or a specific sample, and that sample was captured at a time, at a place. So all of that metadata will also add up information to explain, okay, what's going on with antimicrobial resistance. I mean, just to have the genes won't tell me anything about if they're being transmitted between two individuals. I will need that metadata information from both individuals to actually describe those similarities or differences or that transmission. So metadata is key. Also, one of the things about databases and what I mentioned, for example, for the context tab, if we only have the bacterial information without the metadata, it'll be like having the number without the name. So yes, there's information, but I cannot connect it really to what my biological problem is. So metadata is key to really be thorough to actually solve that question of how's AMR resistance going on.
All right. Okay. Thank you very much, ma'am. Okay. So my next question is when you talked about how to recover DNA from bacteria, you said it moves, you get the reads out first, and then you move it down to contigs, then to scaffold. So, like, how does it move from reads to contigs? Like, is it that there's something that you have to do to the reads, or I was really blank there.
Sorry, I didn't understand the question. Can you maybe talk a little bit slower? It wasn't clear in the audio.
Okay, so I said when you were talking about how to recover DNA from bacteria, um, the pathway you used, it started with reads, then it went on to contigs, then it went to scaffolds and to genome. Yeah,
Yes.
so my question is how do we convert reads to contigs? Because I didn't really get the parts that like I didn't really get the picture you used to explain the process.
Okay. Yes, that's for example the assembly pipeline, and maybe to describe it in a little bit more detail. In this case, the assembly pipeline, it starts with the reads, but the reads, as I mentioned, there are like multiple copies that might have some uh, overlapped information. And actually, the programs that we use for bioinformatics uses that approach of the overlaps. So in that overlaps, what you think of, it's take together that we have the overlaps on one side, and also take into account that you've got a read that can only be read in one line. So it'll be from the 5' end to the 3' end. So there's an order. They're not just flipped. They will be in just one side. So with that information, you can have one read and point it to another. And that across all of the available millions of reads. And with it creates a graph. And with the graph, you can look for the shortest path, and the shortest path actually gives you its sequence. That's what's called the overlap layout algorithm. So that algorithm is the one that specifically does that process of coming from the FASTQ files, which are small, to actually get like, okay, let's make a staircase of those overlaps. And with that staircase, you will get a really, really longer sequence. Yes, exactly, babe. The or Laplacian consensus approach or breeding graphs. And that's inside the programs, for example, like Spades. So you can use the FASTQ files, use the Spades, and that will give you the assembly as an output. And the assembly would be as best, well, it could be fragmented, but it will have, well, shorter sequences, longer sequences than the original 100. So those will be the complex.
All right. Thank you very much.
Okay, thank you. Michael, you can go ahead, please.
Okay, thank you very much for that wonderful presentation. My question is this: when sequences are obtained from the sequencing machine, you made mention of Illumina. I have used Sanger before, so this sequence came in chromatograph. Is it that when you decide to open it with a specific tool, it converts the file format? Because I initially, when the sequence were sent to me, I attempted to open it using Notepad. It's arranged in a format, probably that was the FASTQ format, I don't know. But later when I downloaded Bioedit, and I attempted to open it, it's arranged in a different format. So does it depends on the format the software you're using to open the reads, or it comes naturally in a FASTQ format?
This question is a little bit tricky for me because I'm not familiar with Sanger outputs. So what I think that they usually are, it should be FASTQ files, but in reality, I haven't worked with it. I've worked with the other technologies, for example, Illumina or Nanopore, and they both give you FASTQ files, but I don't know if in Sanger there are previous files that can also be, well, text files. If you can check if they're FASTQ files, if they have this structure. Maybe the ID would be different because of Sanger. It'll have like possibly the isolate name, and afterwards other information from the Sanger platform. Afterwards, the nucleotide sequence, the plus, and the last line, the quality. And that block of 4 lines repeated for sequence 1, sequence 2, sequence 3, sequence 4. If it looks like that, it most likely it is a FASTQ format. But otherwise, I'll be completely blind about what Sanger or outputs are. I'll have to check that out.
Thank you very much.
Okay, yeah, follow us to it. You can go ahead, please.
Okay, good evening. Thank you very much for the presentation, Ma. So my question is, you talked about Illumina sequencing. So when we get those sequences and we want to do the quality check that I talked about, so what happens if it fails the check, the quality check? Like, is it that the whole library pair Illumina sequencing process is like?
Yeah, well, that needs a further details because, well, to check for the quality of the FASTQ files, we had one of those bioinformatics standard tools that's called FastQC. And the FastQC file has a standard output in HTML that describes with graphs different indicators of quality. So depending on which indicator, you can check when it failed. because it could fail at different points. It could fail because the DNA was, for example, too degraded and the quality of the nucleotides couldn't be as recovered as best, or the reads were really, really small and the sample size, and the reads just got shorter. It could be, for example, that there's contamination, so you can see it, for example, in the peaks of the GC content, or it depends on the different indicators. So the most common ones are, for example, that during library preparation, there wasn't a correct adapter attachment to the sequences, and just adapters appear in the output. So that's like, okay, how do we see it? We've got lots of overrepresented sequences and nearly not enough an amount of reads. And what else can happen? That DNA is degraded. It could happen from, you can see it with the equality that's not the best in during sequencing. So that's on the last line. The first score will be a little bit low. And other kinds of errors, for example, well, the contamination, I mentioned the different GC peaks. And another for contamination would be, for example, seeing that the amount of ACTs and Gs across the whole sequence will be just in several parts of the sequence will be overrepresented. So that's like, okay, now here we have some DNA that shouldn't be there. So it actually, it's to evaluate all the different indices from the FastQC, and from that take a decision. So where the error could have happened. I hope that solved the question. I don't know if there are more questions. Yes.
Yes, you did. And there's something I'm also curious about. Is it only when we get the sequences from Illumina we can like know, like when we want to do the quality check, we can know that it has been contaminated or it's like a bad sequence? Is it that the machine cannot tell us like error that this?
Well, I think that there are a few that can be seen during running the sequencing. Like, for example, the ones that don't have the adapters attached to will just not see anything. Like that sample, it's absent. Others can be actually be be picked up during microbiology sessions. And I'm not an expert on this part, but I've talked a lot with the lab from my team, and they sometimes mention that, okay, if the DNA doesn't have the rightful quality and that they can check with some tools, they will simply not go on to the sequencing. I mean, in this case, for any of these processes, we try to work with the best of the best. So the best quality of the data, the best quality of the DNA, the best possible library prep to get good results. If at one point the data just says like, this is not good, we will have the tough decision of maybe discarding that information. Because the computers will analyze anything, but one rule of thumb, junk that comes in, junk that comes out. So in this case, it will be better to keep the best quality across the whole process. But yeah, in that sense, I think that it'll be good to talk to the lab members, maybe Toby, that can help you with describing like, okay, no, in here we can find the contamination with this and this other different items, because it'll be better to pick that contamination beforehand as best as it can.
Yeah, thank you very much for that. So,
Okay,
Blas,
thank
do
you.
you want to Do you have another question? Okay.
Okay.
Okay, I guess
Are-
not. So, I think that's all we have for now. Although I dropped a link on the group chat, a Google form. So like if you have further questions I can ask maybe later on or while watching the recordings, you can also fill the form and then ask your questions there. And then I would forward it to her, or then if you can address that, then would also to. Okay, thank you very much.
Okay.
Yeah.
Well, thank you for the invitation. I enjoyed it a lot.
Yeah, thank you very much. Okay, so okay, I have another question from Victoria. Please, you can unmute and go ahead.
Okay. So, Victoria, what's your question?
Victoria, are you still there? You are still mute, please.
I actually didn't mean so it was a mistake on my end. Sorry.
Okay, that was a mistake. So for the form, you can also fill the form of attendance and then maybe like add comments if you have any comments about the presentation or any other stuff. You can also do that, fill it. Yeah.
Yeah,
So
okay.
thank you very much,
Um,
and it's very nice having you. And like we have a lot to unpack from this presentation. So yeah, well, thank you very much.
Okay. Oh, you're very welcome. I really enjoyed the making up this presentation. I hope you enjoyed it too, and that it was hopefully not so much information that it will get like too much. But hope this serves you well. I mean, I've shared the presentation with Tochi, so you can share it also if there's need for it. And any questions, well, I might be able to attend them through email if you got any more questions.
Okay. Yeah. So thank you very much. I will definitely keep in touch after this. Yeah.
Okay.
Yeah.
Well, see you in another opportunity, and hopefully you enjoyed bioinformatics too.
Sure.
Okay. There you go.